Okay, so welcome everyone. Thank you, Christian, for coming to give this seminar, and the stage is yours. Just go. Thank you.
Great. Thanks, Thomas. So good morning. Well, it's morning here, so good morning, everyone. I'm going to today take you through some work that I've sort of ongoing in terms of how thermodynamics affects information flow in metabolic networks. So just a quick overview. I'm going to take you through a background and kind of motivation for this work, why we're doing it. And then there's a kind of a story in 3 parts I'll take you through, looking at a sort of simple toy model that we can use to understand information flow in metabolism and constraints thereon, and then understanding that in the context of regulation as well. And then I have some bioinformatic structural type evidence in support of kind of the central hypothesis here, and then I'll take you through some future work as well. But before that, I'll just give a quick introduction of myself. So yeah, my name is Christian Euler. I'm an assistant professor here at the University of Waterloo in the chemical engineering department. So I am an engineer by training, maybe a bit of a different background than others on the call, which is great. So I do systems biology and metabolic engineering in particular. I work with microbes. My main gig is to engineer microbes to make chemicals from waste. But as we're talking about today, I also work to understand the fundamentals of metabolism, stability and evolution and origins of metabolism. So that's kind of the theoretical arm of my research program. And a big part of that is the informational dynamics stuff that we'll talk about today. So getting into that, let's talk about some motivation for that work, why I'm interested in information flow in metabolism. So, the usual conception of information in biology is attached to nucleic acids, right? So we have DNA is sort of a long-term information molecule. RNA is more of a short-term information molecule in terms of extracting, usable information from DNA to make proteins and other functions. But within this kind of idea, the role of metabolism and information processing is underdeveloped. And we know this because there are many, many examples of autonomous regulation within metabolism. So one thing that I learned presenting this work before is that the kind of, in my world, when we talk about regulation, we're really talking about autonomous regulation. So this is happening within the system and by the system, not from some external controller. And there's lots and lots of examples of this in metabolism, which is evidence of information processing capacity just emerging from this complex network of overlapping reactions. But it's not really well appreciated or well understood how that information processing capacity does emerge from this metabolic system or the system of metabolism. And this is broadly kind of relevant or important because the concentrations and rates that occur in metabolism provide information to the cell that then can interact with what we think of as being more traditional informational molecules like the genome. So for example, external and internal states are signaled by various concentrations of metabolites in metabolism. So the ATP to ADP ratio, for example, is a known signal of energetic state of a cell. And then one of my favorites, fructose 1,6-bisphosphate, is a really well characterized flux sensor of glycolytic flux. So the steady state concentration of fructose 1,6-bisphosphate correlates very well with glycolytic flux. So the concentration of this metabolite then provides information about that flux. Guanosine tetraphosphate is one that tells us information about the amino acid pool size, and when that pool size is depleted, the PPGPP signal changes translation rates, controls translation. So not only do we have really good examples of metabolic signals relating to very important information about cell status, we also have strong examples of those metabolic signals interacting directly to regulate some other process, like guanosine tetraphosphate, regulating transcription rates, or translation rates rather. And so a lot of these signals, of course, do feed into sort of genomic interactions where we can have transcription factors binding and changing expression, and then feeding back into metabolism by changing, maybe the structure of the network in response to an environmental change, or changing the concentration of a given enzyme in response to an internal state. So, realistically, much of the information that in fact gets to the genome sort of passes through this filter of metabolism, and that is, I think, generally not really well understood. So that's kind of this gap that we're aiming to fill. So yeah, most information characterizing the cellular state is linked to metabolism, and of course, as we understand, binding is how information flows in cells. So you have to have some kind of physical interaction between a molecule whose concentration may mean something, and for example, a protein. So this is sort of the very simple high-level toy example that I like to show often, even my undergraduates. But if we have a particular state, cell state, in that condition, we can have a certain concentration of a metabolite at steady state, and it may bind an enzyme and regulate the activity of that enzyme through, say, allosteric regulation, inhibition, whatever that may be. And then, of course, in a different state, there may be more of that metabolite at steady state, and as a result, more is bound to the target enzyme, regulating its activity with respect to what's going on in that other state. And so we have to have this sort of actual binding event. Information is physical, and it's very apparent that it's physical when we think about biological systems like this. And where do all these binding events come from? Well, they have to come from stuff that is in metabolism, right? We have to have some kind of molecular signal that can bind to a transcription factor before anything happens. So how does that information get encoded in that metabolic signal is sort of the broad question here. Of course, there are some things, some states where the information flow does bypass regulation, bypass metabolism entirely. So, osmoregulation is an example. There are other environmental conditions in particular that don't require sort of enzyme or metabolic mediated information flow, but the vast majority of them, I would argue, do. And so we need to understand how information gets from metabolism to affecting the state of the cell and providing some kind of control information. So ultimately, this kind of brings us to the motivation. Enzyme regulation, is something that's very well understood in terms of the biophysics. We have strong characterization of where regulators bind to enzymes, how, when they bind, that changes the activity of an enzyme, for example. But there's very little in the way of characterizing how enzyme regulation is organized in metabolism. So why are some metabolites inhibitors, but not others? What characteristics make a metabolite a good inhibitor, structurally speaking, in terms of the metabolic network? Which enzymes are good targets of regulation? So why should a particular enzyme be regulated in a pathway versus the others in that pathway? And also, why is enzyme activation so much rarer than inhibition? When we look at all of the regulatory interactions that happen in metabolism, about 20% are activation interactions and 80% are inhibition interactions. Some of this may, of course, be down to the fact that it's easier to inhibit an enzyme than it is to activate in terms of the structure of that enzyme. But of course, there are examples where activation is prominent in metabolism. Fructose 1,6-bisphosphate is a good example. It activates pyruvate kinase in a lot of organisms across the tree of life. And so why should that happen and not some other kind of inhibitory interaction, for example? So the answers to these questions have implications for the emergence of information processing. So how do we go from a few reactions that are maybe not processing information about their environment to a minimal set of reactions that can process information about their environment? And of course, this has implications for origins of life, the metabolic-first hypothesis in particular. I'll talk about that kind of at the end of the presentation. But there's many other sort of implications here. One that's very applied in my work as an engineer is how do cells evolve to an environment that no longer has a ton of information because it's not variable. So for example, when we grow cells in a bioreactor over many generations, the environment's very controlled. What changes occur in those cells in the absence of environmental information? What do they do to handle that? And that's useful in terms of boosting the production rates of bioprocesses, which is something I'm interested on the applied side. So it's not just a fundamental question. There are sort of applied implications as well. Okay, and so I just want to kind of lay the groundwork to make sure that everyone's sort of on the same page, because we'll be coming back to this kinetic form throughout. So this is kind of the base unit that I analyzed for this particular work so far, which is just a reversible enzyme-catalyzed reaction. So, a substrate S arrives at a rate defined by some upstream reactions, or maybe an uptake reaction, and it's converted to a product P reversibly by an enzyme, which can then leave the system. And so there's very sort of standard saturation type kinetics that we can use to characterize this, which I think everyone should be familiar with. And I just want to make sure that we're again on the same page, where we have the overall rate of this reaction is expressed by sort of a catalytic capacity defined by the turnover rate or the turnover number and the amount of enzyme that's there, but also by saturation factor. So how much substrate is available relative to a Michaelis-Menten parameter here called Ks. But there's also a thermodynamic parameter, which is tells us how far away this reaction is from equilibrium, and the further away it is, the higher the driving force in a given direction. And so this reaction is characterized, or the degree of reversibility of this reaction is characterized by an equilibrium constant, which I think we're all familiar with. And so I really do like to say that all reactions are reversible, and I think this often ruffles feathers, but the idea is that all reactions are reversible, but some are so favorable in a given direction that we'll never see them go in the opposite direction. And we can characterize that with this continuous parameter, which is the equilibrium constant. So it can take on any value from 0 to infinity exclusively. So this tells us how the degree to which reactions are irreversible, and that'll be important later on as we go. In general, of course, if we have a high equilibrium constant, then products are favored, and a low equilibrium constant favors the substrates of this type of reaction. And in previous work, I showed that if we have an enzyme-catalyzed reaction that is very favorable in the reverse direction, that enforces accumulation in an open system where we have flux entering and leaving this reaction. That enforces accumulation of the substrate. which is sort of intuitive. Of course, if you want a reaction to go in a given direction and it would prefer to go in the opposite direction, increasing the driving force for that reaction is one way to kind of move it in the direction that you want it to go. And life does this a lot. Many metabolic reactions are unfavorable in the direction that they occur. A great example is malate dehydrogenase in the TCA cycle, which has a standard Gibbs free energy of, I think, around 20. So it's very favorable in the reverse direction, and yet life drives it forward, and has been doing so for billions of years. So what we found is that because this substrate has to accumulate, and because enzymes saturate, the downstream rates in a pathway like this one that is favorable in the reverse direction become much less sensitive to substrate concentration. And that's a consequence of saturation type kinetics. As a substrate concentration, steady state substrate concentration increases, the slope of the tangent at a given point along this rate curve is smaller. And so small changes to the substrate concentration are not going to have a large change in the downstream flux at steady state. As a result of this, what we found is that this sort of creates a situation where the substrate concentration is a good readout for the upstream rate, the flux. So if there are small changes in this flux, then those changes will be amplified in a concentration, which can then do binding, of course, as we talked about before, and transmit information. And so that idea, we'll kind of revisit that as required, but because of this, the sort of idea here is that if we can perturb the concentration of a substrate of a reverse favored reaction, then that perturbation will not propagate downstream, because the downstream enzymes that require that substrate or are using that substrate are typically operating more closely to equilibrium, and at saturation in that case, sorry. And then, therefore, if a metabolite is the substrate of a reverse favored reaction, it's likely a good regulator because it is a readout of upstream flux. So just going back, the context for those hypotheses are basically, if we have a very reverse favored reaction, the substrate has to pile up in order to get it to go in the direction we want. But if the substrate is very high, then the downstream rates are not sensitive to changes in that substrate concentration. And so we think that those types of metabolites, the ones that are substrates of reverse favored reactions, would make good regulators, good informational molecules, because they accumulate as a function of the upstream flux. Okay, so hopefully that makes sense so far. I think there's time also for questions and clarification as we go. But with that kind of in place, I'll take you through the first sort of part of the story, which is a simple model system, a toy system that we can use to characterize sort of informational dynamics and really in particular constraints thereon. So as before, as I kind of mentioned before, it's very sort of simple Michaelis-Menten type or saturation type reversible kinetics, representative of sort of a metabolic reaction. So in this sense, we have substrate again arriving at a given rate, and the product leaving at a rate as well. So this is an open system. We have flow through and a directionality to this reaction, which means there's continuity as well, right? There has to be a mass balance, and this choice is for the representative system is basically, it's the smallest unit of analysis that we could look at, and most metabolic reactions kind of look something like this. So it kind of gives us a good sense of what's going on across metabolism and at least define constraints. And so for this case, reversible Michaelis-Menten kinetics are sort of similar to what I've shown you before. We have capacity factor, a saturation factor, and a thermodynamic factor. And here, throughout the presentation, I'll call this delta because it's this term as written, 1 minus this ratio, is the sort of intuitive distance from equilibrium. So when it's 0, this reaction is at equilibrium, and other values are away from equilibrium. And so it's a nice measure to just take as a as a measure of that distance from equilibrium. So we'll call it delta from now on. Okay, and the analysis of this system, because it is small, is quite simple. You know, we have in a mass balance, we have two species to look at. We have this kinetic form showing up again, which can be non-dimensionalized very straightforwardly, giving us a parameter k, the equilibrium constant, and some downstream kinetic parameter that tells us how quickly P leaves the system. And in this case, also the upstream flux is a sort of control parameter for this system. So this mass balance has steady state values that depend exclusively on that input flux. And that's sort of what they look like here. So again, this comes down to the fact that there's continuity. The flux that's entering this reaction has to be leaving it. And so we get this very sort of simply defined steady state that really just depends on that flux and is parameterized again by these two parameters, K equilibrium and the downstream kinetic constant. Now the fluxes are valid between 0, so no flux entering the system, and up to 1. This is the kinetic capacity of the downstream enzyme. So if we just go back quickly, the scaling for this upstream flux is actually this kinetic capacity, K cat times E. So we can never get a flux, a scaled flux that is greater than 1. So this gives us the entire range of operation for this particular reaction. And of course, as you saw, the steady state value for the substrate concentration is a nonlinear, has a nonlinear relationship with that input flux. And that relationship looks something like this. So we have kind of this hyperbolic type relationship, and as this equilibrium constant is adjusted, it actually becomes more and more linear. So the black line here is sort of this limiting irreversible case where we have a very strongly forward favored reaction. For every degree of sort of reversibility, we move this curve back, and it becomes much more linear. So what we can see is that for very reverse-favored reactions, strongly reverse-favored reactions with low K values, like around 0.01 in this case, the steady-state substrate concentration is a linear function of the flux, the upstream flux, which is what I mentioned before, that this substrate concentration is a nice readout of a flux, and therefore it might make a good regulator in metabolism. So our goal here is to figure out how perturbations about a given steady state are propagated through a system like this. Of course, this requires that we understand the sort of distribution for species in that system, but this is challenging for even what is at the macro scale a simple system like the one that we're considering today. There are many primary steps involved in enzyme binding, forming a product, product release, etc. So it can get very complex to find expressions or even to estimate these distributions. And so I use a tool called linear noise approximation, LNA, for this. And what it really does is takes macro scale trajectories, which are easy to predict or easy to generate. As I mentioned before, in our case, the steady state concentrations, this trajectory is really only dependent on one thing, which is the flux that we put into the system. And so linear noise analysis can take that very sort of easily determined trajectory and from the system structure generate a distribution at any steady state for the species in the system. And so I'll take you through a little bit about LNA for those who, again, don't know. I never know sort of what people understand or have heard of before on the call. So just very quickly, linear noise analysis is a fluctuation dissipation balance. Due to Alpha and Ehrenberg, you can check out this paper. It's really useful in terms of noise approximation. And so the key things here we have, we're looking for a covariance matrix. This can help us understand informational relationships between species in the system. And then to find that, we need a Jacobian. So this is the matrix A, quite simply defined again because this system is simple itself. So it's relatively easy to get a closed form for the Jacobian. And then we have a drift matrix as well. So the drift matrix is defined as such, where S is the stoichiometric matrix and F of X is a diagonal matrix that, a matrix that contains the rates of the system, and the diagonal of that is just a matrix where all of those rates are along the diagonal. There's zeros elsewhere. So as it turns out, when you kind of go through and analyze the system and figure out what the drift matrix looks like for this particular toy system, it really, again, only depends on the flux. So it's quite simply defined. And this is really useful and kind of makes it quite tractable to solve this fluctuation dissipation balance. So ultimately, as I just showed you, everything is uniquely defined by the upstream flux, what the rate is going into the system and coming out of it. And then, of course, we have some parameters, a couple parameters. So then to find the mutual information between perturbations to S and P, the substrate and product of this reaction, which is ultimately what can tell us about how information is flowing through this reaction, we can go to the basic definition for mutual information. And because linear noise approximation gives us Gaussian distributions as the estimates for the steady state distributions of species in the system, this very simply becomes a correlation or related to a correlation, which can be easily determined, uniquely determined using the covariance matrix that this fluctuation dissipation balance yields. And so this is really what we're looking for here, the mutual information between substrate and product in this system. Now, this is assumed that we know the upstream flux with certainty. That's not really realistic. However, as you'll see, I'm comparing two different conditions with the same flux in this case. And so this is still a useful basis for comparison, even though we sort of assume that the flux is really well known and has low variability. Okay, so the workflow for this, basically there's two things to look at, the fluxes and the equilibrium constants. For every equilibrium constant, there's a range of fluxes between sort of 0 and 1, scaled fluxes between 0 and 1 that the system can operate at. And there's a range of equilibrium constants that are meaningful. So in E. coli, for example, the meaningful range is sort of 10 to the minus 4 to 10 to the 4. Of course, most reactions fall within kind of the moderate or in the middle of that range, but there are some on either extreme. And then so for a given choice of equilibrium constant, the flux can be varied between 0 and 1. The system can be solved, the fluctuation dissipation balance can be solved, and from that, the mutual information can be determined. And then, of course, one can find the channel capacity as well, which is the largest mutual information for a given K equilibrium constant in this case. And so this is kind of just schematically shown here. Vary the equilibrium constant, and for each equilibrium constant, vary the flux, and then find the channel capacity, the maximum information that could be transmitted through this reaction. And so I've done this for a range of parameter values for the system to understand how it behaves. And I'll show you kind of minimal results today, just in the sense of containing everything to a particular story. But what I can say is that these are robust to changes in the downstream turnover rate, which is sort of the only other parameter that we can adjust in this case. So what we see is, first of all, that the distance from equilibrium constrains this mutual information. So when we're at equilibrium, there's a potentially asymptotic approach towards a very, very, very high mutual information or infinite mutual information between up and downstream concentrations in this case, or perturbations to those concentrations. But as the distance from equilibrium increases, we can see that drops down to 0. There's no information that can be transmitted. And the way this sort of works is that the system is pushed to an arbitrary distance from equilibrium for large input flux. So as the input flux increases, depending on the equilibrium constant, that either pushes things further away from equilibrium or closer to it. So the distance from equilibrium is not being manipulated directly. The only input to the system is the choice of flux, and that defines everything else. And so you can see there's this case where far from equilibrium operation means no information transfer, essentially. But life operates far from equilibrium, so this is a bit challenging. We would expect maybe some ability to transfer that information, to take internal states or external states and make use of them. But in the far from equilibrium case, that's not really true. So what's going on? Well, as it turns out, channel capacities for this reaction may actually be quite large, but it really depends on what's going on. So here we go. I've calculated the channel capacity for the metabolic reaction or the metabolic channel over a range of equilibrium constants. And for each equilibrium constant, I varied the flux between 0 and 1, the maximum possible range, and just found the maximum mutual information. And you can see there's some numerical kind of jumping around because this isn't a numerical estimate. So there's some tidying up that needs to be done. But in general, this constraint curve emerges where the equilibrium constant, when it's very, very low, we actually can get a lot of information being transmitted through this reaction. Strong correlation between up and downstream metabolites or perturbations to those metabolites. And this is actually fairly consistent up to a point, at which point it drops down to 0. And given that this is due to the equilibrium constant, this means that it's a hard constraint on channel capacity because equilibrium constants don't change. If you have a given reaction in metabolism, it has an equilibrium constant, and no matter how you change the metabolite concentrations at steady state, that value will always be the same. So ultimately, as the equilibrium constant increases, the channel capacity, the maximum possible information that could go through that reaction, drops down to 0. Now, this might seem kind of extreme. Equilibrium constant sort of is getting quite high here. But this is, again, over the range of reasonable values for, say, E. coli metabolism. And given that pathways are quite conserved, that range is reasonable for a lot of other organisms as well. So reactions with very low equilibrium constants have the highest information carrying potential. So these are reverse favored reactions. And as a result, these reactions sort of structurally have high information loss potential, because you can imagine that if under some conditions you could get very high information transfer from such reactions, and then when those reactions are driven far from equilibrium, that information disappears. That may be a problem if you're a living system that needs to get information from the environment or information about internal states. And so these reactions are sort of seem to be places where you might expect some kind of a mechanism to capture that information that could be lost. So quick summary of that part of the story. Perturbations to metabolite concentrations are just not propagated in far from equilibrium conditions. It seems to be impossible. And the capacity for informational propagation is dependent on the equilibrium constant. It provides a hard constraint on that capacity, where reverse favored reactions have the highest capacity to propagate these perturbations. And this is what we suspected from the start, because these are the ones that accumulate stuff that can then bind to proteins. And of course, information in biology is tantamount to binding interactions. So, okay, this seems to be a problem. How can it be rescued? Of course, I've got the answer to that question. So in order to look at that, I sort of then moved on to a model system with regulation. So I'll take you through this sort of toy system. The basic unit is still there. So we have flux entering this reaction in the form of substrate S, which is reversibly converted to product P, which leaves again the system according to some turnover rate. So this is the baseline system, from before. But now let's say that we have some kind of binding event with a protein that changes the state of that protein. And so S can bind to, say, a regulatory enzyme, or a regulated enzyme rather, to put that enzyme into, say, an active or inhibited state. Now we have two channels over which information may be propagated. So the solid box shows you what I call the metabolic channel, and the dashed box shows you the regulatory channel. And the question is, when we have a perturbation to this substrate concentration, say due to some perturbation to the internal state of the cell, how does it propagate along the metabolic channel and or along the channel, and are there conditions in which one channel is more effective than the other? My motivation here was really, my PhD, I was very interested in allosteric regulation and enzyme inhibition. So that's why initially this sort of system, I considered it from the point of an enzyme that's regulated. But as we'll see, it doesn't matter. It can be really any protein that binds there and has a change in state. So just to make sure, again, that we're page with what this looks like mathematically, our mass balance now just has one additional species to think about, which is that the activated or inhibited protein or enzyme. And so that just binds according to some kind of, again, binding kinetics and unbinding kinetics, which of course shows up in the mass balance for the substrate, which is the molecule that's binding to the enzyme. As it turns out, at steady state, though, this drops out, and so the steady state for SMP is defined as it was before, but now there's also a steady state regulated enzyme concentration to consider. And that steady state is, again, uniquely defined by the flux, the upstream flux, because that flux defines the steady state concentration of the substrate, and of course the steady state substrate of the concentration of the substrate. The steady state concentration of the substrate defines how much of this enzyme is bound. So now, as before, we have a mutual information between perturbations to S and P, but there's also the regulatory channel to consider. So how does information move along that channel? Well, we can capture that as a mutual information between the substrate and the regulated enzyme. Again, with a channel capacity being the maximum possible information we could get along that channel. And because LNA gives us Gaussian distributions, once again, this it comes down to a correlation between these two variables. And so here's what we see. This is a very similar curve to what I showed you before, where we have distance from equilibrium and the mutual information between the substrate and whatever downstream species we're talking about, depending on the channel. In the solid lines here, we have the metabolic channel, and in the dashed lines, we have the regulatory channel. So what you can see is that no matter which choice of equilibrium constant we make, the the performance or behavior of these systems is exactly sort of inverted. When there's no or little information that can be transmitted through the metabolic channel, the information that is transmitted through the regulatory channel is maximized. And this happens whether we're close to equilibrium or far from it, along the entire operation line for a given equilibrium constant. So as a reminder, each of these curves represent one equilibrium constant, and along these curves, we're just adjusting the total flux moving through the system. So, in some sense, the regulatory channel appears to capture information that cannot be captured with the metabolic channel. And so we might describe it as sort of rescuing information flow, i.e., under some conditions, some reactions would transmit information, but under other conditions, that information is not transmitted, but it can be captured by a regulatory interaction instead. And we can look at that again in terms of how the equilibrium constant constrains things. So this is a very similar curve again to the one I showed you before. We're zoomed in a little bit though, and the black dots show us the trend for the metabolic channel capacity, and the blue is for the regulatory channel capacity. So you can see that the regulatory channel capacity sort of behaves similarly. It's sort of similarly constrained by the equilibrium constant, although it does perform a little bit better kind of in this range, in the higher range of equilibrium constants. But it's ideal to sort of have it maintain, to be constrained in a similar way, because then under different conditions, it can capture the same amount of information or similar amount of information that would be lost. So we, for example, operating here, or if we have a system constrained to be here by its equilibrium constant, the maximum possible information we could get metabolically is around, kind of just below 1 bit. And if we were to lose that, we could capture back 0.6 bits through the regulatory channel. So we want this curve to sort of look similar in shape to the metabolic channel capacity curve, and it does, with some small deviation. So this is really an ideal setup to maintain information about internal states over a varied environment. So even though the environment is changing, that information can be captured because the regulatory channel has a similar relationship to the equilibrium constant as the metabolic channel does. So it's sort of like a backup control system. So it seems like across equilibrium constant values, perturbations are best propagated by regulation when the system is driven far from equilibrium, whether by high or low flux, it doesn't really matter. And the channel capacity of regulation is constrained by the equilibrium constant in a way that's similar to the metabolic channel. So regulation might be organized to ensure that perturbations about the sort of internal state of the cell are propagated regardless of the condition that the cell is in, or regardless of the environmental state that the cell may be in. Now, is there any real evidence for this? This is, kind of been all in silico, all kind of hypothetical development up to this point. It's of course very hard to observe metabolism kind of in action. It's a very fast system. But what we can, the way we can conceptualize this is that it's kind of from an evolutionary lens, which is that we would expect the structure of metabolism to be consistent with this idea that regulatory interactions exist to maintain information flow. So we do that by looking at structural data that's available. And there's various kind of pathways or routes that I pulled together for this. So first of all, I took an E. coli genome scale metabolic model. These models are sort of readily available. They're based on genome sequence data, annotated genome sequence data. And to that, I assigned equilibrium constant values to metabolic reactions using something called Equilibrator, which is just an API that's available to take reaction strings and assign equilibrium constants or Gibbs free energies to them using database values determined from the component contribution method. It's a machine learning based approach. And IJO1366 is just the name of the genome scale model that I used for this work. Now, this gives us the sort of thermodynamic side of things, but then also regulatory and non-regulatory information has to be captured as well. And so there's a database called Brenda, which is an enzyme database, contains information about sort of every enzyme that's been characterized. It's not built to contain information about regulatory interactions, but that information is there for many, many enzymes. There's also the allosteric database where this information is explicitly collected and it's purpose-built for regulatory interactions. So I took all those equilibrium constants, then I looked at all the metabolites in that genome scale metabolic model, and using Brenda, assigned them the status of being regulatory or non-regulatory based on whether those substrates are known to participate in regulatory interactions or not. And so then from here, we can kind of look at how thermodynamics and regulatory status are correlated, at least structurally. So what has evolution landed on after, a few billion years of optimization? So in E. coli, when we look at the distribution of equilibrium constant values, and in this case, I'm taking something called the minimum one. So to find this, we look at every substrate, we look at all the reactions that could consume that substrate, and we find the minimum equilibrium constant. Because we're talking about, the constraining value with this work, that minimum one is the one that makes the most sense to look at. It's the one that's going to constrain how information could flow in theory. But as I'll talk about on the next slide, it actually doesn't matter whether we take the minimum one or not. And so then for all those substrates, we can look at whether they're regulatory or non-regulatory and look at the distribution of minimum or constraining equilibrium constants downstream. And what we see is that there is a very clear difference in the thermodynamics of reactions consuming regulatory versus non-regulatory substrates. So in the non-regulatory case, the reactions are much more forward favored. The median constraining equilibrium constant value is around 17. So these are quite forward favored reactions. Whereas for the regulatory reactions, the median is around 1, but you can see the distribution is quite shifted and it's a little bit tighter even than the non-regulatory distribution. So it does seem like regulators tend to fall upstream of highly reverse favored reactions, as kind of expected. And so I was curious then if this persists across organisms. There's lots of data available in Brenda, and there are many, many genome scale metabolic models out there. So I pulled together the information or the data and sorted it according to, for which organisms there's sufficient high quality data to sort of do this analysis. Turns out there are 6 of them. So E. coli, P. putida, Bacillus subtilis, Saccharomyces, rats, and us, of course, Homo sapiens. So the similar analysis in this plot here in panel A, where we have the regulatory equilibrium constant distributions in purple and the non-regulatory in gold. And no matter what organism we look at, there's a clear difference in these distributions, where regulatory substrates tend to be upstream of reverse favored reactions, no matter what organism you're talking about. Of course, here I chose the smallest equilibrium constant, and I'm not showing. I don't have the data shown for you today, but it doesn't matter whether you pick the lowest value or an average or a geometric average of all of the values of reactions that consume these substrates. The distributions look very similar, or at least the relative differences in them do. In that regulatory reactions regulatory substrates rather, tend to be the substrate of reactions that have low K equilibrium values, so reverse favored reactions. And then you could ask, of course, what is the sort of probability of finding a regulatory reaction given an equilibrium constant or a constraining equilibrium constant? And to do that, bind all of these values from all the organisms across the tree of life here that we were able to look at, and found a correlation between the constraining equilibrium constant value and the fraction of substrates that were regulatory versus non-regulatory, which, that's sort of a proxy for the probability. Interestingly, at a K value of 1, it's like a 50-50 chance. So half of the metabolites tend to be regulatory and half tend to be non-regulatory. And the slope, this correlation is fairly strong. Now, this is, of course, from untargeted data across many organisms, and the binning is as a heuristic, task. So there's some noise. For example, up here we see there's clearly some kind of deviation happening here. And this is really something that when we're working in the bioinformatics world, we do expect. So again, not shown here, this correlation persists regardless of the bin size that's chosen or the number of bins, and it is robust to whether you choose the minimum downstream equilibrium constant or a geometric average or an average of all of the downstream equilibrium constants. So it seems like this correlation is persistent across the tree of life. Okay. So I'll give you kind of my conclusion and perspective, and then I'll talk about next work or what's coming up next. So I am strongly in the position that metabolic constraints are also informational constraints, and I think that that's hopefully an idea I've developed and shown to you here. Information can't propagate at reactions operating far from equilibrium, which poses a real challenge for life as a fundamentally non-equilibrium process. But if there are regulatory interactions by substrates of those far from equilibrium reactions, then the information can be rescued. Okay. So my perspective on this, again, far from equilibrium operation maybe is inconsistent with having information in general. I think that, of course, the talk I've given is really related specifically to metabolic interactions, but we can intuit a more general understanding of this, which is that if you're operating very quickly, you have less information about what's going on versus if you're operating very slowly, if you're operating very far from equilibrium, rather, then there's very little understanding or ability to understand what's going on. And there is, I haven't linked it here. It's kind of work in development, but there is obviously a link between, the destruction of information and far from equilibrium operation versus equilibrium operation and the preservation of information. So now it seems like if we have some reverse favored reactions in the network plus some kind of non-reactive regulatory interactions, then we can capture that information flow, which brings us to the sort of bigger picture, which is that in order to have some information about an internal state, and therefore to be able to reconcile this against a goal like persistence, some of the reactions participating in that internal state must be reverse favored. So when I think about this as an engineer, we would like cells to grow as quickly as possible in a reactor for us so we can make a lot of stuff very quickly. And cells in the environment are also trying to grow very quickly because they are competing against other cells that are growing. And so one might think that just going very, very quickly, having a lot of very far from equilibrium reactions that are operating very quickly might be the way to win, in terms of being a living, evolving system. But as it turns out, it seems like in order to have robustness or even some information about internal states, some of those reactions have to be ones that don't want to go in the direction you want them to go. And so from a metabolism-first hypothesis for the origin of life, this kind of gives a coarse-grained or macro-scale understanding of how information flow can happen without an information-carrying molecule like DNA or RNA. And so I think that that's relatively interesting. In terms of the next step with this work, I didn't show today this or talk about today these results at all, but in building that network regulatory interactions to analyze it, or sorry, rather in collecting all of those regulatory interactions to analyze them, I inadvertently built a network. And so then the question was, is there any structure in that network and is it interesting? Well, as it turns out, there are some feed-forward loops in the metabolic regulatory network that are highly overrepresented. So these are just the various canonical feed-forward loops we could have. And you can see the excess counts. This is relative to a randomized network of most of these are kind of near zero. We don't see any excess of those feed-forward loops. But for the incoherent type 2 and incoherent type 4 loops, we do see an excess. They show up a lot more than we would expect from a random network. The incoherent type 2 is all inhibitory interactions, so we might expect this just kind of spuriously from the fact that regulatory interactions and metabolism tend to be inhibitory. But the incoherent type 4 involves two positive or activation interactions. So this is very, very interesting. And we haven't characterized this yet at all in terms of their functionality and how they preserve information, maybe how they how they can preserve information over wider states, or we don't know. So characterizing these loops is kind of next on the docket. And then there's also a question around sort of the channel capacity of an entire pathway. So the linear noise approximation approach is quite tractable in a pathway with many linear interactions or linear reactions rather. It could be relatively easily applied. Of course, the matrices get bigger and bigger and bigger, but their contents don't get any more complex. So realistically, you could, for example, find the channel capacity of glycolysis in a similar way. And so then the question is, are there informational properties related to the conservation of these pathways and metabolism? So have they persisted because they are robust informationally, for example? And how do, in particular, I'm interested in how do information dynamics and stability intersect? So in life, we have this idea of autocatalysis, which I've kind of shown here. In cells fed acetate, they use an autocatalytic route to make more cells and energy. And these autocatalytic systems are sort of inherently unstable. So is there some stability value in being able to, for these systems being able to measure something going on inside, i.e. having information at the metabolic level about those reactions? And so in general, both of these works require expansion of the framework beyond a single reaction, as I've shown you here. And so that's broadly the next step, but I'm very curious to hear your thoughts and if there's other ways to go with this as a next step. So with that, I will take any questions you may
have. Thank you. Very, very interesting. Great. So let's come back for a moment. I wonder if anyone has questions. We have like 10 minutes for discussion. So you, San, you want to say something? You're muted. Let
You're
me
muted.
unmute you.
Just a very general comment. This was absolutely fascinating. I think you're doing really important work. I couldn't follow all the detail. I've downloaded your 2022 paper. Is that the closest to what you presented, or have you got something new?
So the 2022 one is a good starting place, but the paper for this is in the review pipeline right now. So I'll share it with Tomas when it's out, and he can spread it around. But unfortunately, it's not published yet. The reviewers are also having trouble following all the details, as it turns out.
Yeah.
Yeah.
But I mean, there is a huge meta level implication, and it's wonderful that you have all the detail.
Yeah.
Because there has been lots of argument and really not until the last 20 years or so with developmental biology was there understanding that evolution was not just genetic, and a whole kind of resurrection of Lamarck for having anticipated this long before we knew about any of these mechanisms, of course. So I think there's a really big implication to your work.
Thanks. Yeah, I mean, I agree. I think what happens, what seems to happen in biology and historically is that a discovery is made, and then everyone kind of focuses on that way of understanding things, right? In particular, we're talking about metabolic regulation here. Feedback inhibition was like, it was discovered, and then it was like, well, that's just the way metabolic regulation happens, is that we have substrates that are then feeding back, or products that are then feeding back and inhibiting. But as it turns out, that's a real small subset of all of the regulatory interactions that exist. And so, yeah, I think you're right that there's been kind of a much of
Yeah,
focus on
I'm
the
even talking more
DNA.
generally about the whole epigenetic,
Yeah,
right?
yeah, exactly. So, yeah, so hopefully, again, hopefully the reviewers can understand this so it gets out, so people can start to think about these things in that context as well. But yeah, certainly the 2022
Well,
paper
maybe
is
you could
a
do a
big step.
simpler version for all of us.
Yeah, yeah. I do. There's also another one called or
Saw
sorry,
that one.
proteome partitioning constraints in long-term laboratory evolution. That might be of interest to you, Zan. This is one where I was a contributor. My collaborator here, Matthew Scott, we looked at the Lenski experiment where they evolved E. coli or still are evolving them. And what we found is that there might be the loss of an information preserving interaction that has allowed for much more evolution in those cells because they're in a consistent environment where they don't need to capture that information. So that's one you can also look at to kind of get a bigger picture view of things. Yeah.
Okay. So I want to understand better if in the short time we have, but then we will meet up again for sure.
Sure.
The idea of information flow, just to clarify to everyone. So it's how you go back to the first diagram,
Yeah.
when where you were showing like the purple and I think green products like reactants and products, and if I have more of these and I change that, so that one,
Yeah.
the previous one even, or the next one, I think.
Oh, this one?
No, the one, the other diagram, the one with that one.
This one. Yeah. Yeah. So, and this is where the 2022 paper will
So
be.
what is
Yeah.
information here
Yeah.
for making, like, because information, it's and typically it's a bit, it's a state of a certain device
Yeah.
that will be in one or another true or false state, and that's a bit of information, and that's what most people are accustomed to
Yeah.
understand information, or a letter, so that's why the DNA, it's information and we all get it. But now this is like information through the dynamics, right? So it's the
Yeah.
it's the flux, is the flux state the one that determines the information, right?
Effectively, yeah. So,
Yeah. So
yeah, that's
please
right.
elaborate.
Yeah.
I'm only
So,
up to pedagogical, for pedagogical reasons. Yeah.
yeah, yeah. Yeah, so you're absolutely right in saying that like DNA is like just very intuitive how information, because it's literally a code, and that's what information theory originally sort of came to describe,
Yeah.
or was originated. Yeah, so here the idea is that the flux uniquely defines the steady state of this system. And from a well, backing up, we can ask the question, information about what, right? There's information about anything, really. And what is a cell interested in knowing about? Okay, so that's kind of, and when, it's anthropomorphizing, but when we talk about evolution, what information is useful to persist is maybe a question that we could talk about, right? So the external state is information that would be useful to know. And the internal state also of the cell would be useful. So the internal state of metabolism is defined by a distribution of fluxes, so rates going on. And in particular, whether those rates are
Wait.
valid. Yeah,
Because these rates are also defined by the concentrations and the kinetics.
that's right.
It could
Yeah.
be but either way, either you look at the kind of cause, let's say, the concentrations and the kinetics, or the consequence, which is the fluxes. Well,
So
I mean, it's just to sort in one way. But then you are looking at the
yeah,
fluxes as the main determine, because a given flux can be reached by two different collections of concentrations. Like, I mean, 1 times 2 or 2 times 1, they both
yeah,
will give you the same rate. So
yeah.
that state is equivalent for in this perspective.
It's only equivalent in terms of the Michaelis-Menten equation we write down on paper, but we have to recognize that um, there's also a protein constraint for cells. So that's something that's not that haven't thrown in here at all. But
My last
the,
question is about PDE. How can we make it with PDEs rather than with so we also have to put the space and boundary conditions and
yeah,
yeah.
yeah.
So
Yeah. So that's in the expansion, right, of this framework.
of course,
But
technically speaking, but keeping it more philosophical yet
yeah,
still. So the flux is the information state of the metabolism. That's what this interpretation is so far like advancing, like, or an
yes,
hypothesis, let's say a methodological principle.
yes.
Okay.
I think it's a very a broad hypothesis, and the justification for that is that to a cell, all that actually matters is rates, because let's talk about a cell in the environment where it's competing against other cells for stuff. The ones that can get that stuff the fastest and use that stuff the fastest are going to persist. And
Yeah.
so we often think about, and binding happens as a function of concentration. So there's a disconnect there. We often think in concentrations, but realistically, cells need to balance many, many different rates in order to grow very quickly. So yeah, the sort of inherent perspective of this work, and maybe this is something that should be communicated earlier when I'm communicating it, is that the fluxes are the most, information about fluxes is kind of the most important thing for a cell from a metabolic perspective. And so the flux defines a steady state concentration, which then can be used for binding. And binding is how that information becomes meaningful, right? How it becomes actual information for a cell, because a flux is a calculated value that a cell can't directly measure. So there has to be some kind of associated binding event that allows that to be measured. Does that kind of make sense?
Yeah, it's starting to make sense to me.
Okay.
I said that for pedagogical purposes because it's your mindset, and the traditional understanding I have is like we have a state, the state defines the flux,
Yeah.
and there is this game. So where is information there? And now I
Yeah.
think
So
I'm
so we can
following better.
imagine that let's imagine the cells at a given steady state, but that state is now perturbed. There's some kind of environmental perturbation or something along those lines. We can kind of think like what happens first, right? So the first thing that happens is you would expect a perturbation to some substrate or some metabolite concentrations. Enzymes take a long time to make. So if there's an environmental perturbation, of course, eventually we can expect that the enzymes, the transcription translation is going to start doing its thing, and enzymes will change. So the fluxes will change because
Yeah.
because the enzyme
So
concentration change.
propagate.
But kind of the first thing that happens is, of course, there's going to be changes to some of the concentrations of the metabolites. So how does that information get captured, right? Because the transcription translation stuff is actually downstream of that. The signals all originate from metabolism for transcription translation to change. So that's the idea here is if in this type of reaction where it's very forward favored, if this flux were to be perturbed, actually there would be very little change in the concentration of the metabolites here. And that's because there's a lot of capacity in this enzyme to handle any changes upstream. So if there's a slight increase in the substrate concentration, the enzyme can will naturally respond because of saturation to increase flux downstream, and we don't expect a big change in the substrate concentration. But in reverse favored reactions, if there's a change in this flux, then there should be a really significant change in this concentration because the downstream rate can't change very much because the enzyme
No,
should be near
this
saturation.
this part I got.
Yeah.
So it's also about reading or storing the information of the environment, internal and external information, as you say. So
Yeah.
this gives a lot. Well, we have only like -1 minute ahead. So I wanted to ask you one more question
Sure.
about at the end, in the conclusion, you said non-reactive interactions.
Yeah.
Was an advice in a conclusion somewhere at the very end,
Yeah.
and I
Yeah. Yeah.
didn't understand that. What do you mean by non-reactive interaction?
Right. Yeah. So I should use that language throughout or not at all. So what I mean by that is just a reaction like this one, like the regulatory type. So this is not reactive in the sense that S is not being transformed in any way. It's binding to a regulated enzyme and changing the state of that enzyme to be active or inactive, let's say. Whereas the reactive interaction is like a substrate binding to an enzyme and turning into something new, undergoing a reaction. So that's what I mean here. Basically, that having
So
some regulatory interaction.
I misinterpret this. So I thought that S turns into E star. No, S just remain S, and
Yeah,
ER turns into E star, and then by reconnecting with S can go from E star to ER again.
that's right. Yeah.
So
So
S
it's
is
just
the catalyst then.
no, I wouldn't describe it that way. S is it binds to this protein, whatever it may be. So it could be an allosteric
Yeah.
effector, for example.
No, in my understanding, a catalyst is it's a species, as a molecule, a species that has is reactive, is participating in the reaction, but with zero stoichiometry. That's my definition.
Okay.
But it's not
In that definition,
maybe
yeah, sure,
under
yeah.
that definition. How would you call that?
Well, that I think to me is a bit too that captures in biochemistry what we might also call a ligand. So
What?
a ligand, which is what I would call S in this case. S is a
Uh,
ligand of ER,
like,
because
yeah.
from my understanding of catalysis, a catalyst has to do a reaction, and so this is just say this S is binds to ER, maybe at some site, but it doesn't. Neither of them chemically change, right? So it's bound, but it can unbind as well, and there's no chemical
But
change.
then S and E star is a compound molecule.
It's a complex,
Let's
yeah,
say spatially they become one molecule. They
yeah,
become
sure,
one space unit. Okay. So then, yeah.
yeah.
Okay.
Does that make
So it's
sense?
a reversible. I mean, in my view, it's a reversible kind of moleculization reaction. So S
Sure.
can become SE. I will call it SE, and then
Yes.
back to
Yeah.
E and S separate. Okay. Yeah.
Yeah.
So all this space dependence also is to study further, because there might be also spatial structures that facilitate or prevent from this, let's say, non-spatial description dynamics to be modified, perhaps,
Yeah.
because
I
yeah,
think generally when it comes to metabolism, we assume um, that things are well mixed and that there's, especially when we're talking about like bacterial metabolism, that everything can interact with everything else on a concentration dependent way.
Hmm.
Of course, there are compartments when it comes to eukaryotic cells, right? And so there may be spatial limitations, and some of these interactions may exist in some compartments and not in others, and
Yeah.
that is also interesting. There's some evidence that, for example, mitochondria shuttle, they get rid of malate particularly because it builds up in order for this reaction to go forward,
Mm.
and that may be detrimental in terms of these regulatory interactions that malate's involved in. So yeah, there is a spatial element when it comes to eukaryotic cells anyways to think about.
Yeah. Okay. So, but still, it's applicable to bacteria without. I mean, this
Without
is
consideration, yeah.
yeah. Okay. Awesome. Well, we are past 5 minutes, but this
Okay.
was great. Very, very interesting, and there will be more people watching this later. I will upload this video to YouTube
Yeah.
tonight or tomorrow, and we stay in touch, Christian. Then, okay.
Yeah, absolutely.
So
Okay.
stop the
Thanks all.
recording.